Hi, my dear students. This is Dr. Jayanti, Associate Professor, Department of Biochemistry from MGMCRI, Puducherry. Here I'm going to take glycolysis. So, from the glycolysis word, you can refer glyco plus lysis. So, glucose breakdown. Glycolysis is nothing but glucose breakdown, and it can be called as Emden-Mayer pathway or EMP. So, university exam they'll be asking or describe the pathway of EMP with energetics, regulations, and clinical significance. So, first we'll go for definition. Glycolysis, breakdown of glucose to pyruvate under aerobic condition or lactate under anaerobic condition for energy, which is nothing but ATP production. So, where it occurs? Tissue site, all tissues of the body. It is the only pathway that is taking place in all the cells of the body. Mostly it happens in cytosol. Cytosol is nothing but an organelle, subcellular organelle, in which it has a clear fluid portion in cell, subcellular cell. So, starting material, glucose, and end products, pyruvate as well as lactate. So, you have to remember glucose converted to pyruvate under aerobic condition and lactate under anaerobic conditions. Now, we'll start the pathway. So, you have to remember three reactions. One is investment phase, second one splitting phase, and third one production phase. So, first we have to invest and then split off. At last, production of ATP will be there. So, in investment phase, glucose converted to glucose 6 phosphate. Glucose 6, which is nothing but aldo compound, converted to keto compound, fructose 6 phosphate. Addition of phosphate with fructose 6 converted to fructose 1,6 bisphosphate. So, now investment phase over. In investment phase, glucose 6, fructose 6, fructose 1,6 with the help of enzyme hexokinase or glucokinase, phosphofructokinase, phosphohexoisomerase. So in this PFK is rate limiting enzyme. Along with that, hexokinase and glucokinase are very important rate limiting enzymes that takes place in all the cells as well as in liver. And next, splitting phase. So fructose 1,6 converted to glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate. This reaction will be interconverted. And next, dihydroxyacetone phosphate converted to glyceraldehyde, and this glyceraldehyde will be converted to 1,3 BPG, 1,3 BPG to 3-phosphoglycerate, 3-phosphoglycerate to 2-phospho, and 2-phosphoglycerate to phosphoenol pyruvate, which is the end product of glycolysis. So, this phosphoenol pyruvate plays an important role in converting pyruvate with an enzyme enolase. And this enolase will be inhibited by fluoride. So, that's why for diabetic patients, we have to take fluoride coated tube in order to avoid false results. So, in this case, aerobic condition, pyruvate will be converted. When there is lagging of oxygen, when a person is climbing up the hill or during starvation, if it is prolonged, pyruvate converted to lactate. So lactate, if it is converted for a longer period of time, it may causes lactic acidosis. So here the enzyme is lactate dehydrogenase. So thus the reaction is over. After this reaction, you just remember ATP molecules. Altogether 10 ATP molecules will be produced. So in that 2 will be invested. So 10 minus 2, 8 ATP molecules will be produced for each glucose under glycolysis. And you have to remember inhibitors of glycolysis: iodoacetate, arsenate, and fluoride. And next, significance, main important thing is energy production. In muscle cells, glycolysis provides even under oxygen deficient conditions. In erythrocytes and lens, anaerobic glycolysis is the only pathway that provides energy, as these cells lack mitochondria, because TCA cycle and beta oxidation, two major pathways that provide energy that takes place in mitochondria.